it's a kind of a very informal thing we are doing here. It all started when we wanted to talk more with the Brazilian guys in Australia, like Osmar, Luis, and Diego, and Alexandre Siqueira. So we started doing this thing, and then I thought, why not? We're starting to go really abroad, and so it's nice that you are starting the first one really international. I mean, it's the idea is to talk a bit about your work in general, what things you like, and you can explore one or another paper or idea you have, and you can introduce
Okay, so
the students there. It's up to you. You can
great.
show stuff on the screen.
Okay, so following your advice to talk a little bit about where I come from scientifically and all that, so I did prepare with a few slides you sent me before, a short presentation, like talking about my view about diversity and stuff like that, and where I come from. And
Great.
then I thought it'd be nice to talk about the paper, Itai Granot's paper in GEB, and he can present his paper, and then that'd be a good place for discussion, perhaps.
Great.
Okay?
I think it's a good
And
idea.
and there are quite a few people from my lab here too. So I don't know if you want to do an introduction or something. It might be hard to do online, but and yeah. So from my what I see here, I see Mai and Shachar and Tal. From
Okay.
Mahmoud, Itai Granot is going to talk about his paper, and Shira Salinger, which is a master's student, and
Great.
and Merav. Okay, so quite a few people. So I think I'll try just to start and give an overview, like I said, and then hand over to Itai to talk a little bit more. But detail, we can just open it for a conversation. I'll try to
Yeah.
be okay. Try to be short. Let's see. Okay. Now, can you see me? Can you see the presentation?
Yeah. Yeah.
Okay. I can't
Okay.
see anybody actually, except for my presentation. Is there any way to can see myself here in the
Yeah.
presentation?
You're
Is that too much?
you will be solitary now, but we are all here.
Okay. So I don't see anybody but just my presentation. Anyway. Okay. So like I just said, to talk a bit about myself and where I come from. Okay. So before I told also that I'm quite busy because I'm working on a grant. So he actually did a lot of work for me, and he found all these pictures of both of us and Kadu and other people from a long time ago. So I'm going to present the pictures Sergio found. So this is I've in France in 2013. So we were there for a gas power working group meeting talking about reef fish diversity gradients. And Michel hosted us very nicely in France. It was fun. And more pictures also with Diego. And this is a meeting in Israel. So the following year we met in Israel. We had a small grant to look at patterns and processes in coral reef fish abundances. And that was a lot of fun. And many people in the lab were in this working group. And there was with Seb and also with Nicotelli. More pictures from around Israel. So that was a lot of fun.
It was great times there.
Yeah. It was great. And so just so you know, people not familiar, so this picture over here of the group is in the beach of the Red Sea. Okay, so down right below the beach there are coral reefs and reef fish and lot of fun. And the picture over there is a Dead Sea. So there looks very similar, but there's no fish at all. And no fish diversity to work on. Okay, so that's just a bit of a picture of memorabilia, but now going back to science. So I just want to give a bit of scientific background of where I come from scientifically. Okay, so as an undergraduate, I did my degrees in Ben Gurion University, and they're very much focused on mechanisms of coexistence. So the pet animal is gerbils, rodents, and many people like Avramsky, Bert Cutler, and also my later supervisor, Yaron Ziv, really try to work on these species and understand how they coexist, if it's via temporal partitioning or food partitioning, or different mechanisms of habitat selection and data dependence. And I spent most of my undergraduate years and also later on working at nights, running across the sand dunes in the desert, looking for rodents and trying to capture them and count them and stuff like that. And it was a lot of fun. But in the end, I felt like it's really just too local. So we're working on coexistence, but we're not really understanding the larger scale patterns.
Yoni,
So
just one thing.
yeah,
Speak a little bit slowly so people
okay,
can get you better. It's
right,
all okay, but just a little bit slower.
no
Thank you.
problem. So after working on these local mechanisms of coexistence for quite a while, I started my master's and my PhD, and I looked for something else. So I understood this can't make sense. We can't look so local. We need to look at larger scale processes. And then I came across these plots of the local versus regional richness plots. Okay, so the Y here is where I have to see is local richness, and the X-axis is regional richness. And these are things that Bob Rickliffe started, and later Howard Cornell picked up on. And the idea is that regional richness really determines local richness. Okay, so regional richness is on the X-axis, and what you get to the local scale really depends on what you have on the regional scale. And the regional richness could be affected by things like evolutionary processes, speciation, large-scale plate tectonics, other things, and they trickle down to affect local scale richness. And this idea in general really intrigued me, and I decided to shift my focus into these large-scale patterns. I'll take the time I have here and also just talk a little bit about this particular pattern. So when you look at local versus regional richness relationships, there are basically two ideas. One is having a complete straight line, an unsaturated relationship. And the idea is if you go to different places in the world, you might have different regional richness. If you have also local richness that is higher, and places are regionally enriched, it means that there's no local interactions. The more species you have at the regional scale, the more species you have also at the local scale. And a different approach would be, a different pattern would be a saturated pattern, okay? So that's an idea like that, okay? But you have more and more species at the regional scale, you have some kind of cap, some kind of limit to the species you get at the local scale. And this was used to infer about local interactions, okay? I have your mistake. So saturated means that the local interactions that actually limit the number of species at the local scale. And when I started, this seemed like a really nice idea to actually separate patterns from processes. So we look at patterns of saturated versus unsaturated relationships. Maybe you can say something about the processes or the importance of local interactions. Now, this was later proven to be wrong, okay? So you can't really infer anything about local interactions from these patterns, but they still can tell you a lot about how regional and local processes interact. Now, just as a side note, every few years, I would say every 5 years in ecology, there's a new idea or a new pattern, a new method that we can say maybe we can use these patterns to say something about the processes underneath. So in the past, people looked at species abundance distributions, right? The log theory, the log normal, or that some kind of other something coming from the neutral model to say something about the processes that structure communities. And it was very popular for a while, and then people understood that many different processes can cause these patterns, and it's not so good. And then people looked at phylogenetic and functional community assembly. For example, if the community is functionally over dispersed, maybe it means something about competition, right? And everyone's really excited about that, from the first papers of 2000 of Webb and others, and people started looking at their own community to see if they're over dispersed or not. And very fast or very soon, people realized that actually many different processes can cause an over dispersed pattern. But don't really say too much about processes. And more lately, people are really excited about co-occurrence patterns, right? And they use things like joint species distribution model, JSDM, to say something about the probability of co-occurrence given environmental factors, and if any residual correlation exists, to say something about interactions, predation, competition. And I think people are more and more realizing that's also not robust way to infer processes. So in the end, I think that in macroecology, you're kind of doomed to be in this loop of excitement about a new method to infer processes. But after a few years, we understand that it's not as simple as we think. And the good thing about that, we usually learn something on the way. So when we do this, we get really excited about new methods. We learn something on the way. We realize it doesn't do what we wanted to do. We do learn something about ecology as we go. And I think the important thing to understand is that we can't really infer patterns from processes in a straightforward way using one or two methods. So the only way I think, okay, in general, you can really infer processes if you look at multiple patterns, 4, 5, 6 patterns that have different predictions, and then put them together to really understand what processes are important. Okay, so going back to what I did in my PhD. So my PhD was to really look at areas that really differ in the regional diversity and see how it affects patterns of diversity at small spatial scales. So I sampled reef fish across different areas of gradient of diversity. We all know about this reef fish diversity gradient. Most the highest richness in the coral triangle, and it goes low when you go west and east. The sample in the Great Barrier Reef, which is close to the hotspot, more or less. And Tanzania, that has medium diversity. And the Red Sea, that has actually low diversity. And the main conclusion from my PhD, I think, was that regional impacts are very strong across all spatial scales. So when you go to areas that have higher regional richness, you see higher richness at all spatial scales, even, but maybe excluding the smallest spatial scale of a single coral head. That's where you don't really get very strong regional effects, but all other scales you do. So this graph here is the number of species at each of the scales in the Red Sea, in Tanzania, and the Great Barrier Reef. And you can see there's higher richness at all spatial scales when you go to rich and richer regions. Now, that's generally true that regional richness affects local richness, but the pattern also is different for different parts of the community. Okay, so take species that don't have strong local interactions. You can compare them with species that are resident, see different patterns. Find species higher diversity origins. For resident species, they get more or less some kind of cap at the local scale for resident species. And of course, the effect of region on local diversity really depends both on the strength of the regional effect, but also on the local environment. Okay, and this is already moving to my postdoc with Walter Yates at Yale. And here we compared the effect of regional richness versus environmental impacts at different scales and for different groups of species, bird, mammals, and amphibians. And we found that there's a strong regional effect, but also a strong effect of environment. So they interact in a way, affecting local richness. So just in general, so we know the regional effects, we know the local effects. How do we put it all together? So I think there, in general, there are 3 options, okay? So one option is that you have this process hierarchy, that some processes affect the regional species pool. Out of these, and you know this figure, right? Out of these, some species disperse or immigrate, then they're filtered by abiotic condition, the local environment, and then they're filtered by species interactions. Now, the advantage of this kind of flow chart is that if we define the regional species pool, okay, we can use that as some kind of null model and then isolate the effect of what we want to look at. For example, I isolate the effect of only species interactions that we take into account as a given the species pool. And this allows us to actually separate or isolate the separate effects of interaction versus dispersal versus regional processes. I would say that's only one option. I think other options, first of all, is that community assembly is what's really important. These local interactions actually scale up and they can also shape the species pool. So not just species pool affecting the local species composition, can go also the other way around, okay? And of course, the way these species are assembled may depend on the abiotic conditions. So you have a two-way interaction, species pools affecting local communities, but also local communities affecting the species pool. The conventional wisdom is that species interactions are very local and therefore they don't really scale up to affect the species pool, but I'm not sure it's completely proven. And one attempt to look at it from 5 years ago, looking at woodpeckers in North America. And here we looked at interactions, taking into account environmental effect and dispersal effect. And we tried to look at the scale in which these interactions manifested. So in the part over here, we see the coefficient of the interaction, so how strong interaction between species affects co-occurrence probabilities, okay, at different grain sizes. Okay, so 1 kilometer, 2 kilometers, up to 50 kilometers. And here the coefficient is quite strong at the local scale, okay. Gray and the black are positive and negative interactions. And it goes down as you go up in scale. So the effect of interaction seems to diminish with spatial scale, which is expected in a way. We show it here, I think, nicely. We also show that even at 50 kilometers, there's still some signal of interaction. So from this paper, the take-home message is that species interactions do scale up to larger scales, but they become weaker and weaker, and it seems unlikely they will actually affect the size or composition of the species pool. Now, apart from these two options, I think there's a third option that's very frustrating to me, okay, and that's the idea of continuum of scales. That's, again, an idea by Bob Rickliff, and the idea that local communities, okay, are basically, I'll just read the quote, the local communities cannot be used to test hypotheses concerning the origin, maintenance, and regulation of species richness either locally or regionally. So the idea that we don't have a local community that's embedded within a regional community. We have actually individuals interacting with species across many different scales, across the whole species range, okay? Because different individuals from the same species interact with different species in different parts of the range, okay, at different spatial scales over the whole region. And that means the first idea of this very nice flow of regional diversity affecting dispersal, being affected by abiotic filter, being affected by community assembly, might be oversimplistic. Now, the problem with this idea is that it's very hard to know what to do about it. So in a way, I think it's probably most realistic. So that's probably, this is the way probably real communities work. But it's very hard to understand how to actually go about studying communities when they work across all different scales and all the different, spatial temporal scales. But to sum this up, I think that we're still very far from understanding what affects biodiversity. We're making good progress, but I think we're very far from understanding it. We can't really understand processes completely from patterns in a simplistic way. It's going to be very hard to do. And finding, and in any approach we take, finding what determines diversity is really dependent on spatial and temporal scale. So we have to take into account both the large-scale processes and the small-scale processes to understand what's happening. So that's what I had to talk about in the first part. I'll be happy to take questions, and if not, I think we can hand over everything to Itai Ganot, who wants to talk about his paper, which is also related to scale and community.
It would be great if you want to. Yeah, it would be good. Do you have
Itai, go ahead.
Any the figures of the paper to move on?
Yeah, Itai, you have the presentation ready, right?
Yeah, sure. Yoni, I have a question. If you can, again, briefly, the three options you mentioned in I had some technical issues in
So I think one option, of course, is that everything is top down, okay? So you have regional richness, and then the filters of dispersal, and then you have filters of abiotic filters. So if a species doesn't fit into an environment, it won't be seen, right? And then you have the community assembly filters. You have interactions such as predation, competition. And the idea is that they kind of operate one by one, right? And then in the end, you get what you see in the local scale. So you have this nice set of filters. That's one option. I think it's very popular nowadays. The other option is everything goes bottom up, okay? So you have the local interactions, and the local interactions also shape what you have in the region, okay? So you have species competing over resources, and the species excluded would be excluded both at the local scale, so the regional scale. It will affect also the regional species pool, right? So all scales are affected by this local bottom-up process. And the third option, I think, is most complicated. That species are affected at all scales simultaneously, a continuum of scale, right? So you have these interactions affecting things at the local and at the regional scale together, and species are not adapted to a local community or adapted across the entire species range. They need to interact with the species where they are now, and species might be 1,000 kilometers away from them, other side of the range, other species. But as a species, they need to be able to interact with those also, right? They have much more complicated than the first two scenarios, which are a bit more simplistic. But like I said, I'm not really sure how to translate the third scenario into something that's testable and predictable, and you can use it to say something about ecology. So I think most people take one of the first two approaches and don't go into the third one because it's just too complex.
Okay, thanks. I wonder what do you prefer? If I have a 5 minutes structured presentation I presented a year and a half ago in an IBS conference, and it's almost a final result, or I can just open the paper and briefly skim and talk about in 2-3 minutes about the gist of the paper.
Sergio, your microphone is... Turn your microphone, Sergio.
I think it's worth doing the 5 minutes, and then we can go in different places of the paper. Maybe you can tell us some behind the scenes how you came up with the ideas to go with that. But maybe it's worth starting with the 5 minute thing.
Okay. I'll start with the 5 minutes, and I'll try to make it less informal than it was. Let me see how I.
Yeah, you can go slower. Don't need to go in just 5 minutes. Take your time.
Thanks. Let me see. Here in Israel, we mostly work with Zoom. So this is the first time with Google Meet.
In the Google Meet, you have just your right side, you have presenting now.
Yeah, I know. So I'm just looking for the open window to, yeah, great. And yeah, it's also, I'm under Tal Gavrieli, as I had some issues in my computer. Okay, can you see another presentation?
Yeah.
Great. So I gave this provocative title for the presentation, which came first, species richness or specialization. And from the title of my paper, that is niche width and species richness correlation, it implies that we are mostly... Can you see my cursor also, by the way?
Yeah, I can see.
Can you see my mouse? Okay. So it implies that the paper mostly is about this edge of the triangle that you can see here on the correlation between niche width and species richness. But it's actually much more about this triangle, okay, this 3 components, also including latitude, as many times the correlation that is a perception about the correlation between species niche width and species richness and niche width is in the context of latitude, okay, under the common perception that tropical species are more specialized. It's a common explanation for how come we have more species in the tropics. Now, this edge, of course, is unquestionable, the latitude species richness edge, and the edge of niche width and latitude is over the past years, there's growing evidence against this supposed correlation. And in 2004, Vasquez and Stevens, that's, if you want a bit behind the scenes, I think this is like the starting point for this paper. Well, not from 2004, but the paper from 2004. They talked about, they conducted the first review of this correlation, the correlation between niche width and latitude, okay, the questioning the common perception that tropical species are indeed more specialized. And they aggregated 19 studies, and based on these 19 studies, they didn't find support for this correlation. And what they did find support is some indirect correlation through species richness. And as far as we know, the correlation between species richness and niche beds was not yet reviewed. There was no meta-analysis. So this was the first motivation for this study. So this is what we wanted to do, and I searched the literature for all studies that report this correlation. So in other words, I'm talking about studies that report the community level niche breadth. So basically a niche breadth of a group of species in a specific site for at least 3 sites, and also the species richness at that site. And then we either they reported it or I calculated the Pearson correlation between species richness and niche breadth. Ideally, we also gathered the data on the latitude niche breadth correlation. Now, the data here is not up to date. It's, here you can see that I report 50 such studies. Eventually, we ended with 56 and 35 such studies of latitude niche breadth, and eventually we ended with 38. So that the data is a bit different. And this is here in the presentation here I reported that both correlation were found significant. Eventually, after adding more data points and thoroughly thoroughly searching, what's the word for it? Going over all the data again, we found that the correlation between niche breadth and species richness is strongly is significant and strongly negative, even stronger than what's reported here. But the latitude niche breadth correlation is not significant. It's positive, but it's not significant. So this was the first motivation of the study. The second motivation was, and also it was across taxa and ecosystem. You can see that in the y-axis is the effect size. So in other words, the correlation. It's a Z transform Pearson correlation. And here in the left panel, aggregated the 50 studies according to their taxa. As you can see, all of them basically show a negative correlation between species richness and niche breadth. And on the right panel, you can see the same thing with ecosystem. So we had, again, these numbers are not final numbers from the paper, but basically something like 13 studies on freshwater, 33 on terrestrial, and 4 on marine, and all of them showed the negative correlation between niche breadth and species richness. So here we move to our next motivation, and that's I think the more interesting part of the paper is to try and understand. Okay, we have this correlation, but what is the underlying process? And we confronted, I would say, we confronted 3 hypotheses regarding the underlying process. Mainly, there are 2 of them are well established, and one of them was proposed by Vasquez and Stevens in their paper from 2004. So the first one is a more large-scale biogeographic climatic process that was already suggested by Jansen and MacArthur in the 70s. In the Jansen's epic paper about why mountain passes are higher in the tropics. So the idea here is the mechanism that overall more stable conditions in the tropics, okay, low seasonality and also more stable conditions over in geological timescales enable species to specialize in specific environmental conditions, followed by allopatric speciation, and then we end up with high species richness. So here, the niche breadth comes first, and niche breadth causes high species richness. Another option is that it's a more local scale community assembly process. It's mostly mentioned in the works of Rosenzweig and Diamond. And here, let's take species richness as a given, either caused by latitude or by any other maybe altitude, any other effect that can cause in a local area that will have high species richness. Then through competition will force these species to be more specialized, and then it will force them to be more specialized either in their environmental requirements or in their functional role. Okay, so it can be in their diet, or it can be also in their specific habitat that they find suit to their needs. I didn't here in this presentation, I didn't mention the third one by Vasquez and Stevens, so I'll mention it. I'll just say that Vasquez and Stevens also suggested a third option. They called it the asymmetrical nested interactions. So the idea there is that since extreme specialists tend to interact with extreme generalists, so when richness increases, the mean, if we would calculate the niche width based on the number of interaction of each species with other species, the mean niche breadth would increase faster, would decrease faster. So if richness increases, specialization increases faster. So therefore, this is their explanation for why we see in high species, in areas with high richness, we see more specialized species. So that's the third option. So in order to try and differentiate between these three options, we described 4 predictions based on the attributes of the studies that we collected. So the first prediction talks about the first prediction. Let me 1 sec. The first prediction is that under the climatic process, we expect to see stronger correlations in studies that were conducted over larger latitudinal scales. Okay, so if we have a study that is from southern Brazil to Northern America, we expect to see there, maybe that's not a good example because we went over to the other side of the equator. But anyway, as we have a larger latitude, we expect to see a strong correlation between a stronger correlation between niche breadth and species richness. While under the community assembly process, we don't expect to see such a pattern. Sorry, I didn't mention the first prediction here. So the first prediction is about which one is stronger. Sorry. So is it the richness niche breadth correlation or the latitude niche breadth correlation? Again, under the climatic process, we expect to see that the latitude niche breadth correlation is stronger or at least equal to the niche breadth species richness correlation. And as I already mentioned, we didn't find any significant latitude niche breadth correlation, but we found a richness niche breadth correlation. So the first predictions points towards the community assembly process. The second prediction is what I described about that under the climatic process, we expect to find, if the climatic process is indeed the more dominant process, we expect to find stronger correlations in studies that were conducted over larger scales. And again, I'm wrong. I'm sorry. Let's finish with this prediction. And we didn't find such a effect. As you can see here, the effect size on the y-axis and the latitude extent on the x-axis, and we didn't find any effect of the latitude extent of the study on the strength of the correlation. Okay, so that's another point towards the community assembly process. And now I'll go back to the third prediction. The third prediction talks about that under the community assembly, we expect to find a specialization either environmental specialization or functional specialization. Competition can cause both. But under the climatic process, we expect to find mainly environmental specialization. So quite conveniently, the 56 studies that we gathered were separated almost evenly between both of these types. So I think 27 of the 56 used functional niche breadth measures, such as diet or the pollination interactions, and 29 studies have used environmental, such as habitat breadths. And as you can see here in the graph, again, the y-axis is the effect size, the Pearson correlation, Z transformed. And then the x-axis, we have all the papers and all the studies, and they're separated to functional on the left and environmental on the right. And as you can see here, the dashed line with a gray area around it, the 95% confidence interval, there is no difference between the strength of the correlation between functional Nischbett measures and environmental Nischbett measures. So again, it's in favor with the community assembly process. And I think in this structured presentation, I finished here with having all three, because it was only 5 minutes away. I focused only on two hypotheses and not on the nested interactions suggested by Vasquez and Stevens. And I also focused on these three predictions, and these are the conclusions: that we found strong negative correlation between niche width and species richness, that we found that between these two processes, community assembly sees as a more dominant underlying process, and overall, in congruence with what Vasquez and Stevens have found already, it seemed that any correlation between latitude and niche breadth is indirectly through species richness. And here I might jump maybe to the paper and talk a bit also about there was a force prediction. Just one sec. Sorry for that. I'm with Tal's computer, and it's here. Can you see it now?
Yep.
Yeah. It's
yeah. Actually, basically here, and there's the whole gist of the paper is presented here. The three processes and the four predictions, and the one prediction that I didn't talk about in the presentation there is the prediction about the grain size. Under the community assembly, the grain size means what is the size of the site in the study. We have studies that a site was 1 meters of quadrat. I don't know if they sampled soil invertebrates, and we have studies that the size of the site is as countries. So it's Russia and Canada and stuff like that. So it's a really varied. It's a really big gradient of grain sizes, and we hypothesize that following what Yoni talked about before about importance where biotic interactions are playing a key role. So we hypothesize that under the community assembly, we will find stronger niche breadth species richness correlation in more small grain size studies. So basically, in studies that we would find an effect of the grain size on the strength of the richness niche breadth correlation, and we didn't find that. But we feel it's not a really it's not a strong caveat, because under some of the assumption here is that the niche breadth is measured in the site level, but in all of the studies, or in 99% of the studies in this meta-analysis, it's not correct. The niche breadth is measured in the species level. So it's basically, it's not, if in a study there's data from 10 sites, it calculates the niche breadth of each species across all 10 sites, and then averages per site, depends on the presence of which species are in that specific site. So because this is the case, we feel that it's much harder to see such this prediction that we expected to see. If we would have data on the site-specific niche bets, then it would be a really strong prediction. And if we wouldn't find evidence for such a prediction, it would be a really caveat for our conclusions. But because in almost all of the studies in this meta-analysis, the niche bet is calculated in the species level, so again, if you said about a bit behind the scenes, it was like it was a prediction that we added in the last minute. We said, oh, maybe we will find it. We didn't find it, but since we checked it, we feel obligated to put it in the paper. We can't not talk about stuff that we found. But and I definitely stand behind my words that from the beginning we didn't think it's going to be a really strong prediction either way. We find it the no difference between functional and environmental niche bet correlations, and the fact that we didn't find correlation between latitude and niche bets, but found a strong correlation between richness and niche bets, we find it a much more a stronger evidence towards the community assembly process. And in a bit high level, I would say that it's quite, in my opinion, it's quite interesting, and maybe, I don't know, quite striking compared that to the Johnson's Johnson's idea of the mountain passes are higher in the tropics is quite a accepted perception, and here we didn't find evidence for it. We find evidence for that that local processes such as biotic interactions have quite a dramatic effect on large-scale biogeographic patterns that we see. I hope I'm not too exaggerating here. Yoni, if you think I am, jump in and tell and say something. And that's it, I think. I hope it was clear. And if you have any question, I would be happy to discuss it.
Great presentation.
Thanks.
Yeah, you have a few questions over here. I see in the text.
Okay.
One was about the diversity of ways to measure niche breath. So what can you say about it? I think it's comparable across different taxa.
It's not even within the same taxa. We have a really it's a really diverse. It's again behind the scenes. It's a really frustrating process to conduct a meta-analysis, even though eventually it I think it rewards. It was a lot of from paper to paper. I encountered more types of niche bet that I suddenly had to. I realized that I need to try to think whether exactly like what you're asking, whether they are talking, whether they are on the same scale, whether they're comparable. Eventually, I think there is quite a diverse variety of niche bets, but since there's no bias, I think, in the way there's no autocorrelation between, let's say, taxa and specific ways of calculating niche bets. So some of the paper. By the way, there's one mic that is on. I don't know. So, for instance, you can find niche bet measures such as a proportion of extreme specialists in insects and in birds, and you can find a diet breadth in a binary using I don't know category scale in the mammals and also in the other type of taxa. So there is a diverse ways of measuring niche bets, but I didn't find any autocorrelation with one of the predictors that I checked. There was no differences between the Eltonian as a functional and the environmental type group of studies. So overall, I don't think it affected in some directions results. And indeed, in general about niche, but of course you can there is ideas that some species that are specialist in one axis might be generous in a different axis. That might be correct, and if that's the case, then I don't know. It's the I'm trying to think whether in some way it could affect, maybe through publication bias, that I don't know. I in general, I'm not. After doing the process, I had a lot of difficulties of of thinking about. There's also there's not only the issue of how to calculate niche bit, but there also have which index and which measure. But there's also other types of aspects that were more disturbing, in my opinion. For instance, whether they take a niche availability in account, or they take the sampling effort in account. And those issues that, in my opinion, were more problematic, we also regarded them in the text. We also checked. I added predictors on each study, whether they took niche availability into account and whether they took the sampling effort into account, and checked and made some sensitivity analysis based on this data. There's someone is in the mic open, so I wonder if you're hearing me or the car driving there.
No, it's okay. We are hearing perfectly.
Okay. So did I answer? Did I? Who asked this question, Yoni?
Hudson asked the question.
So versus marine versus terrestrial signature, we had really a small amount of marine studies. I think out of the 56, we had 4. So there's no strong signature that we can deduce from this comparison. What are the metrics to assess climatic process in the model? So I think again, it's basically this prediction. It's the prediction that under the climatic process, we expected stronger correlations in studies over larger latitudinal extents. We expected to find at least a strong enough latitude niche bit correlation compared to rich niche bit correlation, and we didn't find any. We found a positive correlation, but not significant. And also the differentiation we talked about between environmental and functional niche bit types, that we quite conveniently, we had 27 studies like this and 29 studies like that, and there was absolutely no difference in the strength of the correlation. So it's all based on these main 3 predictions on the attributes of the studies that regards to the correlation. So this is, I think I'm answering Kadu's answer here. If I'm not, just jump in and ask. But what it takes in consideration, diversity of environment characteristics only, and is it fine to compare among distinct taxonomic groups? So as to taxonomic groups, they're all. We tested it. We tested the taxonomic to see whether there are differences between the different taxas, and we didn't find any such differences. But anyway, it's incorporated in the model as a covariate. So any differences that are based on the distinct taxonomic groups are related. The first part of the question, but what it takes in consideration, I'm not sure what does that relate to, and diversity of environment characteristics. The first part of the question, I'm not understanding. So if you want to ask,
Tol. Here is Hudson. Did the question. I was wondering, like, for many of us, we go to the field and collect data there about the species, about communities. So I was wondering, like, what would be like basic variables for us to collect in order to calculate the niche breadth? And how could we calculate the niche breadth since we like go to the field and we collect information about the species, for example?
So as it's actually related to the first question about how do we actually measure niche breadth. There is quite quite diverse types of how to measure it. And the most basic, if we'll talk about the two most basic, one environmental and one functional that we have. In the environmental, we have habitat. So it might be a habitat categories, boreal forest, tropical forest, and so on and so forth. And just mention which habitat this species was found. Was spotted, and later we can take all of the occurrences of the species or the abundance of the species in the different habitat categories and incorporate them together to niche breadths using such a metric, such as a coefficient of variation or some metric of inequality. If you find all of the species in just a tropical forest and you don't find it in any other area, so you can say it's a specialist to tropical forest. But if you find it in even abundances in different habitats, then it's more of a generalist. And the same thing about diet. You can either from the literature or take gut contents and to see what is the diet of species, and again to see how to try to formalize how many species this species is dieting upon from all species available in his area. And this is, I would say, the most known niche breadth indices. And there is there's more recent options that talk about, for instance, species co-occurrence. A species. So again, just gathering data about species from the field and which species co-occurred with other species. And the idea here is that species that co-occur with many other species, we would refer to them as generalists, while species that co-occur with only a few species will be referred as specialists. And there is more of these I'll say, I'll call it sort of sophisticated options to calculate niche breadth. But back to your question about what to gather from the field. So it might, if you're it might be just describing the field. It might be gathering some environmental data. I don't know, the pH, the salinity, the stuff like that. This can be gathered from the field, but it also can be afterwards obtained from all sort of data sources that can provide this data. And if you're interested in the more in the functional role, so yes, I think it requires some making about which to interact with. Like in plants, you need to be in the field to see and which plants the bees are landing and pollinating them and stuff like that.
Linear model. Have you seen that?
Yeah, it seems. Let me see. I'll look at the graph again. See if something should have occurred to me, and I stopped the. Can you see it? I'm not.
You can see the PDF now.
Okay. Yeah. So let's see the latitudinal extent. I don't see here any other relationship, quadratic or something. So if you see, I would have a relationship. Just talk about this graph. I would say that only around we have effects, but in general, I agree with the comment that if it's stuff that are not linear, you should fit the right model. But in here, I don't see, in my eyes, I don't see any other relationship.
Okay, you have a lot of points over the axis. It's around 45 latitudinal degrees.
Yeah, here.
And that it might, your confidence intervals are quite narrow. But it seems like you have a lot of points over that line, around 45 degrees. And then I was wondering if you adjust some nonlinear model, and perhaps you could find some effect of latitude.
Yeah, but I wonder what is the biological reasoning behind it. Like, why would in a study that expands 45 degrees of latitude, we would expect a strong correlation, and then in a study that expands 70 degrees of latitude, we would expect again a negative. I understand. Like, maybe if I would fit here a quadratic, just mathematically it would fit, but
okay, but you have some breakpoints in some key environmental factors. Even latitude, like 40 degrees latitude, you have a very, for example, a very large variation in temperature and perhaps other factors. Even the area of the continents, you have a breakpoints in the environmental factors, and I was wondering if this effect you find this nonlinear factor would represent some breakpoint in a key environmental factor. Just wondering about that.
Yeah, it's a point. It's interesting. I just want to that it won't just to make it clear that it doesn't mean when you see here a study that it's in around 50 degrees, it doesn't mean that it's expanded from the equator to 50 degrees latitude. It could be also from 20 to 70 or from 30 to 80. Okay, it's
not centered in zero. It's like a
exactly. It's just the extent of the study. So if it's a study in Australia, in I don't know, Australia, maybe it's 5 degrees of latitude, so it would be here. From it's the extent of the latitude, not centered around the equator. So I see what your point about that we have the break in around.
Okay, sorry.
No. That's actually a good point, and it's said that we needed to create a different graph to run a different. That's a moment. I just talked to myself. Sorry, you have me.
Need to read carefully, much more time to get it all, because it's a lot of things going on there, but very nice. Congratulations for you guys. But Yoni, not only based on this work here, but in general, do you think, because you guys are generally including marine and terrestrial stuff in your studies in general, not separating them, but do you think processes are okay to compare marine and terrestrial stuff? There's no fundamental thing to put them apart.
Yeah, personally, I don't think there's anything really different between terrestrial and marine systems. I think many people, when they say terrestrial, they mean they work on mammals or birds, and I think birds and mammals might be different than fish, but it's not that different between terrestrial and marine system. It's more a matter of size and mobility and stuff like that. So, of course, in marine system, you have the dispersal stage of the larva. I don't think it's very different conceptually from the dispersal stage of plant seeds, right? They disperse also in a very broad way. For example, by wind. I think plants and fish are maybe more comparable than fish and birds, for example, where dispersal is more directed in birds. So I don't think it's a marine and terrestrial divide. It's type of species and the dispersal abilities and something about the way we sample them. I think the lab sampling bias is the marine system, right? The way we look at the system is different. And that might cause differences, but I don't think there's any fundamental ecological difference between marine and terrestrial systems. But that's my hunch, right? Not based on data, just my general feeling. I mean, of course, the marine system is three-dimensional and all of that, but I don't think it's a real basic difference. Ecology is ecology, man.
Yeah, okay. Good to hear that. On related to this study Itai just showed us, so you're now more, in general, you're more convinced of community assembly impact on the structure of things?
You asking me or Itai? I don't know, but
both. But maybe in this case, you. Itai is already convinced. He published it. I mean, but in general, I mean, what? Because you showed the different things people started doing along the time in ecology, and community assembly was one of them. But now in your paper,
yeah.
you're stronger on that.
Yeah, so I think when I say community assembly, it's not like we talked before when I presented my part in the beginning, the adoption number 2 was that species interactions are very strong and they affect the regional species pool. So I don't think that's what's happening here, right? What we're having here is that when you have richer areas, the species have to kind of divide the resources among themselves, so they become more specialized. So it's a different approach. So it's not to say, so community assembly in this context is congruent with the idea of like, hierarchy of scales. So there's more species at regional scale that also affects the local scale and they act together, right? In a way. But it's not, it's just saying it's not only climate, it's not only large scale process affecting specialization, but it's actually a richness that's forcing species to specialize.
Okay.
Maybe I didn't.
And any more questions, people? Questions on that? You can write. You can get the microphone. People in Israel are all okay with that. You don't have any questions? Just the Brazilians.
Well, they heard this before, different context. So maybe I ask you, Pedro, what do you think about large scale versus small scale processes and how they affect diversity?
For me, this one, Yoni?
Yeah.
Okay. Yeah, no, I'm still struggling with that. Which processes are operating in different things. I like this question a lot, but yeah, I still don't have general answers because I mean you guys went through this meta-analysis approach, which is nice. And I'm much more focused on the marine stuff that we don't have that many studies already. I mean, to do something like that. Uh, but yeah, it's hard. I don't have an answer. I think we could start. This is something I would like to talk to you later. To try to maybe it's about time to try to assemble things here and there in the marine realm and try to at least to put things even if it's not a complete synthesis, but to put questions in perspective. I think it's something that we should think about. I was wondering that, but we'll talk more specifically later. And but Yoni, at some point when our paper on Journal of Biogeography appears, maybe it would be good for you to present it for everyone again in a few weeks.
Sure, but again, it's either me or Itai that will present it again, unfortunately, because Agav, she's out of science now, so and Itai helps with final analysis. So
Yeah,
it'll be
no,
the same presenters.
no problem.
I could find a different paper somebody else who can present, maybe.
Yeah, it was a pity that the last meetings we did it all in Portuguese, so I didn't um, invite you guys for Diego's thing, but at some point we can put it all together. And Diego today he could not come to the meeting, but he likes this kind of scale studies a lot.
Yeah, so if you have more meetings in English, we'll be happy to join, of course. And I do think it would be nice maybe to have a better introduction of the people. So now, I think we don't all know each other, so maybe it'll be good next time to each one can have two minutes and present themselves.
Yeah, at least you know where it came from and what is the line of research. It will be good. You can start at your side.
Okay, so yeah, so maybe just for then, maybe everyone could just present themselves just in one or two sentences. So you know me and you know Itai, but all the rest maybe can go up one by one and just put the camera on and see who they are, what they're working on.
Yoni, just a question for all of the issues. So, can you read the question there? Just to search, you guys have done any temporal approach?
Yeah, so I have a few things with using time series. More local, we have local trial data, and we're looking through time, what happens through time, and we have nice data that we looked on in terms of depth change that Shachar is working on, in terms of local versus invasive versus native species, which Chezi has been working on. So we have quite a few interesting questions like that. And we are looking at the idea of invasion, to have all the species pool of invasives in the Red Sea, and trying to understand who's going into the Mediterranean. And people are looking at from different angles. So there are quite a few in the lab that are working on this.
History of the rabbit fish in the Med?
Yeah.
That's crazy.
So I want to go first. She's working on rabbit fish.
I heard rabbit fish and I better jump in.
Oh.
Hi, my name is Daphna. I'm a master's student And I'm working actually on rabbitfish and grazing behaviors and testing whether the predator's presence impacts their foraging behavior, and using that as a way to support the theory that MPA can mitigate their foraging. So that's in 2 sentences.
So that's awesome. At some point, I will ask you to have a meeting like that, so about your work, because you have a lot of guys working the brewery. So it'd be nice to have a talk with you.
Sure, happy to.
Let's plan for that.
Yeah, we're also working on that with Seb, Sebastian.
Nice, very nice.
So it's yeah.
So Serge, is your
Sorry, I think everyone will present yourself, okay? A little bit.
Yeah, just go ahead. Anybody? I see Shaka Haikin in front of me now, so it's your turn.
Yeah. Hi, everybody. Well, the main topic of my PhD is patterns and drivers underlying fish depth distributions. Although I do have one chapter which is more local scale about bathoids in the Levant, in the East Mediterranean. So this is my main subject.
And now I see Shira. Your turn. Oh, she's off. I don't know. Okay. Can hear you, Shira. Shira? Okay, so let's go. Merav, are you on? Merav, Omai.
Can you hear me?
Yes, now.
Hi, this is sorry, my camera is not working. So I've just started my PhD, and there are several optional topics, but we'll work on differences between larval fish community structure and adult community structure, and some scale dependence, but we're still working on how getting it all together.
I see Sarah and Shira, but their work is not working.
You're aiming to go to the Mediterranean side or the Red Sea? Or
Was this a question for me? So both. I think both. We'll see how different it is.
But you have data for the Red Sea, and we have data from the Mediterranean. It's going to come soon, and I'm sure you're going to analyze those, right? So, yeah.
Yep.
Shira, want to try again? Okay, so Sarah, I don't know if you can hear her, but Sarah Ochayun, she's you can't see her video now, but she's working on spillovers from MPAs, trying to understand to what extent fish from the MPAs go outside of the MPA, and he's also doing it using actual acoustic methods and sampling, but also doing meta-analysis at the global scale, and that's really cool results. So we hope to publish soon in that regard.
And Shira.
Yeah, we can't hear Shira. I don't know why we can't hear her, and we can't hear Merav either. And be happy to hear the people from your side, Sergio.
But what Shira is working with what?
Okay, so Shira, you can't hear, I'll talk for her. So she's working on looking at core current patterns across the Mediterranean, taking a field surveys and looking at core current, and from them building interaction networks, and to see whether the interaction networks seem to differ between MPAs and outside of MPAs, and from warm to cold waters, how the interaction. So it's very, very related to the ecography paper about geography of interaction that you just published. Okay, so there are quite a few similarities, the approach and the questions.
So, okay, we can start from our side here, just for them to have a quick idea of what we are doing here. So, Lucas, you can start. Lucas is just about to finish his PhD. What it is about?
So hello everybody. My name is Lucas. I'm, as Sergio told, I'm finishing my PhD right now, and I mostly work with trophic ecology of reef fishes, generally butterfly fishes, and that's it.
Anyway, Lucas has one paper about to appear the next days. I will send to you all. It's a nice one on butterfly fish co-occurrence all over the world using that data set. Mostly that kind of data set we used in our Journal of Biogeography that Diego compiled for. So we used that co-occurrences of butterfly fish. It's a nice one. But I will send you as soon as it appears. It's about to appear.
Yeah, we'll be happy to get it.
Oh, who else? Hudson, what you are working with, man?
Hi there. I'm a collaborator of Sergio Cadu and many other friends here. Like I'm a researcher fellow of the California Academy of Science. And I usually work with a lot of stuff about fish, but it's mostly fish from oceanic islands or from mesophotic coral ecosystems. So I'm very interested in biogeography, in evolution, in ecology, sometimes we visit places where we don't have any idea about diversity in general. So we record species, collect new species, describe species. So I'm more about the systems than the specific process.
Great. Who else? Juan Pablo.
Hi everybody, I'm Juan. I'm from Colombia. I'm working in reef fishing and sandwich in Oceania Islands. I have interest in working for cleaner interactions in the world. And now in this moment, I postdoc in the University of São Paulo and work for climatic change and understand the transition zones in fish and the Atlantic different zones. I with sharing my site, maybe you can see my works and my publications in this specific place.
Cool.
One.
We have somebody here who's not a master student, not over here today, but he's working also on time series from data sets from all over. But now it took the Morea data sets that you probably know, so working on that, looking at stability in time series.
Wow, that's nice. I have another student also that work. She's Fernanda. Fernanda's not here today, no, but she's here. So Fernanda, could you tell what you're working with? Nice to know that they are working with time series too.
So hello everybody. I'm working with a temporal series of data in Santa Catarina, Brazil, and I'm looking for patterns that's how can I say? I'm trying to understand if the change in temporal series is related to climate change or changing temperature. basically that.
So climate change or changing temperature, or like the different things, or you mean the same thing?
Yeah, because changing temperature occurs. There's a pattern, and climate change remains to, how can I say?
Yeah, to be proven. Yeah.
Yeah.
Who else wanna talk a little bit what you're doing? Carol, you wanna say what you're working with? She is undergrad working with. Can you tell people?
Hi, everybody. I'm an undergrad, as Sergio said. Everybody can hear me. I'm working with the diet and foraging behavior of fish, Abdeftus saxatilis, here in the coast of Brazil, in Arraial do Cabo and Santa Catarina. But I'm also helping Lucas with his PhD and working with diet of fishes in Trinidad Island. So I think that's it.
Okay, great. Who else is
From
Mara?
my side, I see Chezi didn't talk. Chezi, do you wanna say something?
Yeah, go ahead.
Does this work?
Yeah.
Oh, great. So currently I'm doing my PhD. I'm on my 4th year. My current project is about functional response of fish and the factors that shape the functional response. Things like temperature, activity level, those sort of things. But my main focus is on species invasion, biological invasion, working on a community effect, the size spectra, or native exotic richness relationship, these sort of things. My next project is about quantifying invasion rates from first record data, also time series and more mathematical approach. This is what I do. Nice to meet you all.
Yeah, nice to meet you. Samara, do you wanna talk a little bit? Samara is a master student of us here in the lab. At least maybe I can. Samara is working with something that's nice and has to do with stuff we are talking today. Is going from small plots. We have that small 2 square meter plots with GoPro cameras. So we are trying to understand how accumulating these small areas, how if you change for instance that functional indexes of let's say of course you are increasing some indexes on diversity of functional groups, for instance, but in terms of if you are not all indices respond the same way as you are increasing areas.
Scale dependent, yeah, the scale dependent changes for
yeah. We are trying this kind of thing and trying to do some no models to compare. But this will be something I would like to talk to you later. Also, Ioni, something interesting, but we have to show you more details later on. Who else? It's here. Everyone would just jump in and say, Larissa, what are you working with? You're intending to.
Can you hear me?
Yeah.
Well, as you said, I'm still working on the idea of my project, and so far my overreaching goal is to evaluate cetaceans' functional in a basin in Brazil, and understand how ecosystem functions of this cetacean community.
Nice.
Nice. Mariana, wanna say what you're up to?
Hi, hello everyone. Can you hear me and see me?
Yeah, I can see you. Yeah.
Mariana is now a professor in South Brazil, Yoni.
Congratulations.
Thank you. It was nice to hear you guys talking about the paper. Awesome work. Nice to meet you, everyone. I'm a researcher based in South Brazil, in Santa Maria. I'm next to Argentina and Uruguay, so in the last state here in Brazil, farther south. And I'm basically interested in macroecology, have been doing work from a functional perspective, and also working with historical ecology, trying to understand how were Brazilian reefs in the past, basically going back, far back in the past, and trying to compile data on Brazilian reefs. So these are my main interests at the moment, and I have a lab with several students here, PhD, master students, and lots of undergraduate as well. So that's what I have been doing lately. That's it. We can talk further on another meeting on functional diversity, maybe. That would be great.
Yeah, that'd be really nice.
Yeah, it would be nice to. I think we are promising, and I think it would be really good to have more meetings. That's the thing. Well,
great.
And I was part of the Gaspar group as a PhD student in France in almost 10 years ago. Wow. Yeah. So it was nice to see the picture with you guys and Michel Kubik and Valeriano.
But Mariana, you never overlapped with Yoni at the meeting?
No.
Okay.
We were not in the same meeting, no.
No, and
but of course
yeah, and the meeting in Israel was later on. I was finishing my PhD at the moment, so that's it.
Okay, and another link is that Yoni was on the board of the PhD of Osmar Luis, isn't it? For evaluation.
Yeah.
Osmar, where are you? You're already. Osmar, I think he gone from the meeting now, but that was nice.
So I actually also need to go now to get my kid from kindergarten. But I think it will be great to really have a few more meetings. Like it could be like you said, functional diversity or a different topic, and others more regularly. It'll be lots of fun, I think.
Yeah, no, let's definitely. I think it's worth doing it because it's so much fun to know more about the papers from the author's perspective. It's very nice. Not only reading or reading clubs are nice, but talking with the.
Yeah, but we can also make it more structured. Like instead of just talking about papers we publish, we'll actually think of ideas of analysis because we all have a lot of good data we can use. If you have a really nice idea, we can just actually go out and do it, right? So it could be fun.
Yeah. No, of course we can go from papers to ideas to new ideas and possible collaborations. That's okay.
Great.
Kadu, want to say something?
Just ask Daphne for her email so we can plan a talk about the Ciganos invading everywhere. I like very much the topic.
Yeah, great. So, yeah, I would like to thank you, Yoni, for your time. I know you are in a hurry for a grant.
Just grants, not science, just grants.
Yeah, but we will talk more later on, and we will think about next topics, and we go ahead with that.
That's a fun. Thanks, Sergio, for inviting everybody for this. It was fun.
Okay, Itai, thank you very much.
For people of
Bye.
Thank you all very much. Yeah, joined. Right. Great timing.
Bye.
Okay.
Bye bye.
Bye bye.
Bye bye, guys.